kind of nice hearing about the things that you've done, but it kind of makes you feel older and older. So, yeah. Anyway, so again, my name is Vivek Kumar. I'm an associate professor of biomedical engineering and a couple other things at the New Jersey Institute of Technology. And today I want to talk to you about how we can use biomaterials to present signals closer. Okay, to present signals in a sustained manner in tissue. So, great. So one of the challenges in any kind of drug delivery is making sure that the signal that you present stays localized to the tissue you want it to stay in. A lot of different technologies, a lot of different products that have made it to market, for example, even products from large pharmaceutical companies have used drug delivery scaffolds that rapidly release different factors and which may cause ectopic side effects that where you don't want them, or have a insufficient non-localized signaling. Some of the early work in my lab focused on ocular diseases such as wet age-related macular degeneration, which is basically too many blood vessels on the retina, which obscures vision. So we started out by making anti-angiogenic hydrogels, so gels which kill blood vessels that we can inject into the eye. Now, this is a pretty terrible disease that my dad has even, and he needs to go to the ophthalmologist every single month and get injections into his eye. So the latest and greatest today are injections that you take every 3 months, which is still very inconvenient, painful, and injection in your eye. So what we hope to do was to develop synthetic hydrogels that could either release drug or intrinsically attenuate angiogenesis, needing to be administered once every 3, 6, or maybe even 12 months. So we had some initial early funding on that, made some progress. But what a lot of my research lab has then moved on to and focused on is therapeutic angiogenesis. So instead of killing blood vessels, making blood vessels. So we've been very interested in treating diseases such as peripheral artery disease or the lack of circulation in the peripheral arms, legs, things like that. One of the stents, vascular grafts, large diameter blood vessel substitutes work relatively well, but when the vessels to be replaced are down at the microvascular scale, there isn't a really good technology to encourage blood vessel growth. Now, folks have used gene therapies, stem cell therapies, HIF1 alpha, VEGF, bolus growth factor therapies for over 30 years. But one of the big challenges there is that whatever signal you put in diffuses very quickly and doesn't have a sustained local effect. So we asked the question, can we take growth factor signaling domains and attach them onto material platforms that then become injectable scaffolds that keep the signal where we inject it for a long period of time, treating peripheral artery disease. So a decent amount of what I talk about will be on that. So the material platform that we use is termed self-assembling peptides, SAPs, the top thing there. They have a hydrophilic phase, like you saw at the beginning. They have a hydrophobic phase and a bioactive domain. Now, the hydrophobic regions come together to form beta sheets, anti-parallel beta sheets, and you have self-assembly of these multimers, kind of like the sandwich you see here, where the bioactive domains are on the termini of these peptides in very high density. Now, we can do circular dichroism, FTIR, a couple of different analytical techniques to show that they form anti-parallel beta sheets. But long story short, we form these multimeric structures which have very high presentation of these bioactive domains in green. Now we can add phosphate ions, like in phosphate buffered saline, PBS, and we create these long-range nanofibers that entangle to form hydrogels. So what we've been very interested in doing over the past couple of years, ever since COVID, is trying to understand not just how these materials self-assemble in cartoons, but also how they self-assemble computationally, as well as using computational simulations, as well as novel techniques, novel to this field, techniques such as solid-state NMR. So like I mentioned, as you saw in the previous cartoon, these self-assembling peptides aggregate because of hydrophobic interactions, van der Waals interactions, ionic interactions, electrostatic interactions, but non-covalent interactions, which means these bonds break easily and they reform easily, which means you can syringe aspirate these hydrogels, inject them, and they form a bolus or a bunch where you put them. And the benefit there is that these peptides that form nanofiber, these peptides self-assemble, into nanofibrous hydrogels, like you can see here in this SEM picture, scanning electron microscopy picture, which have nanofiber dimensions on the order of 2 to 4 nanometers tall, 10 to 12 nanometers wide, and microns long. So very long aspect ratio fibers, which we can see through atomic force microscopy over here. Anyway, so these are injectable hydrogels, and I told you about the angiogenic domain that we put on there. It's a domain called QK. It's derived from vascular endothelial growth factor. But in addition to QK, in addition to angiogenic domains, we've put on a number of other domains as well. For example, a dentinogenic domain, which helps dental pulp cells and other stem cells in the oral cavity proliferate. We've added anti-angiogenic domains derived from tissue plasminogen called Kringle. We've made these materials intrinsically antimicrobial, which I'll talk about towards the end of the presentation. We've made versions that can bind and release cytokines in a controlled fashion. We've made versions that bind PCSK9 for cholesterol modulation. And most recently, we made a version that binds SARS-CoV-2, because everyone kind of works on that. All right, so one of the really interesting and cool phenomena that we observed for these self-assembling peptides was the ability for them to bind receptors in a super stoichiometric fashion, right? So for anyone who's familiar with surface plasmon resonance, that's a SPR chip shown in a cartoon. But basically, you take your receptor, you immobilize it onto a substrate, and then you flow your reactant on top, and you see how much binds, and then you flow buffer and see how it unbinds or releases, right? Detaches. So when we flow just the mimic of the binding domains, we see that they bind and they release in a 1:1 stoichiometry, as you would expect or imagine. Now, what's interesting is that when we attach the binding domains shown in green with the self-assembling domain showed in, I don't know, purple, red, well, all these other colors, right? We see that the binding domain has specificity to binding the receptor or the target, and we also have self-assembly on top of the receptor. What this allows us to do, and when we flow a buffer on top, we see that these aggregates, these peptide nanofibers, don't release. So
actually, how do you design the peptide specifically for this receptor ligand interaction?
So I'll tell you that in right after I finish this, right? But that's a good question. So in this particular case, we're using a domain that binds PCSK9, which is a enzyme involved with cholesterol metabolism. But long story short, what we see in SPR, as well as a number of other binding techniques, is that instead of having one ligand bind to one receptor, we have several peptides that bind and self-assemble on top of the receptor, allowing when one ligand releases, the next ligand adjacent to it to bind. Long story short, we have 10:1, 20:1 binding instead of just stoichiometric 1:1 binding. Now, answering Johnson's question, how do we design these peptides? Up until COVID, we went to PNAS, Nature, Cell, all those big journals, and we found out mimics or domains that other people have published that they have found through phage display or that they found through computational design or what have you. When COVID started, we went on lockdown, and then we asked the question, how do we get into the COVID game so that we can get students in the lab working hard? Because otherwise you're not allowed in the lab. So what we did was we took domains derived from ACE2, which binds spike on the surface of the virus. We took domains from ACE2, we computationally, we shortened them, we computationally designed them to improve alpha helical assembly, and then attach them onto our self-assembling domain. We had a simple hypothesis that we can create peptides that bind spike on the virus and then self-assemble on top of the virus, kind of like a fur ball on top of the virus, preventing it from interacting with host ACE2. So to answer your question, for the longest time, we took domains published in literature, but off late, we've started to do de novo peptide design of these binding domains. And ever since SARS-CoV-2, ever since COVID, we've expanded that platform to look at novel de novo binding domains to a variety of other receptors, some of which I'll tell you about. Anyway, we've gotten a lot of traction on the angiogenic platform, and I'll tell you more about that over the next couple of slides. Anyway, so I guess this slide also kind of talks about our design strategy. So the first thing we do in designing these peptide mimics is we take a look at known interactions, so either cryo-EM structures, X-ray crystallography structures of growth factors binding receptors. The growth factor here is an orange goldish color, right? Yeah. And the receptor, in this case, a homodimer of VEGF receptor 2 or VEGF receptor, is in green and blue. I'm not very good with colors, so just work with me. We then look at the specific binding domain, and in VEGF receptor, vascular endothelial growth factor receptor, it's known that the D2 domain in VEGFR2 is important for binding. So we look specifically in that region and we isolate a short mimic, a short portion or piece of the entire growth factor that binds the active site or that binds the binding domain. We then take that mimic, we optimize it based on static and static and dynamic simulations, and then incorporate it into the self-assembling peptide platform. Some of the more recent work we're doing is trying to improve the presentation of these mimics. You can imagine, got a lot of cartoons here, but you can imagine that some of these mimics can be pretty bulky and may not self-assemble along with these beta sheet forming fibers in a structure that mimics how they bind growth factors. To improve that, we're exploring other peptide sequences as well as spacing these binding domains from each other to improve presentation. Anyway, what's really exciting or what's really interesting about this platform, other than the super stoichiometric binding that I told you about last slide, is that instead of having these short peptides that are kind of floppy and bind and let go of receptors, we've designed these self-assembling peptides which stabilize the binding of these mimic domains within the receptor itself, kind of like what these animations show you, right? You got a short little peptide, kind of floats away over 70 nanoseconds. And if you have a self-assembled version of this, it kind of keeps it all in place such that even if one of those mimic domains releases from the binding site, the mimic domain next to it binds to the binding site. So it increases local presentation of a signal. And we like to term that the message is within the medium, right? So within the scaffold itself is the growth factor signaling domain. Now, quantifying this, we've done a couple of different techniques, one of which is surface plasmon resonance, and the other is a technique called microscale thermophoresis. But just to summarize this entire slide very quick, or all these graphs very quickly, we see that QK, or the binding domain that a lot of different folks have published over the past 2006, 20 years, God, wow, 19, 20 years, since it was discovered by the D'Andrea group in Italy. What we've shown is that QK, at least in our hands, doesn't show appreciable binding via SPR onto VEGF receptor 2. It's interesting, this mimic has been used by many people, including ourselves, over the past 20 years. However, no one has shown SPR results. A lot of people show downstream signaling, Western, ELISA for receptor phosphorylation, but no one's actually shown surface plasmon resonance, which we found interesting. So we tried doing it a whole bunch of times, and we too, or rather we did not show any appreciable binding of QK, the mimic domain alone, over a range of concentrations. Now on the flip side, if we attach QK, which is a short peptide which easily unfolds and which is a short peptide, when we attach QK to our self-assembling peptides in a peptide that we term SLAN, we don't need to get into nomenclature, but in a peptide that we term SLAN, we see dose-dependent binding of the angiogenic domain onto VEGF receptor. And as I mentioned before, that super stoichiometric binding, we see that it binds and it doesn't let go. So when we flow the ligand on top of the receptor, when we flow the ligand on top of the receptor, it binds. And when we flow buffer, it stays bound. So it's somewhat irreversible binding. Now, we confirmed this through another technique called microscale thermophoresis. As anyone who works with self-assembling peptides know, they stick to everything, especially plastics. Amphiphiles like to stick to a variety of different surfaces. So there's a solution state version of SPR. It's called microscale thermophoresis. And a specific subset of that is called spectral shift, where you take your ligand, or you take your receptor that you're trying to bind to, you attach a fluorophore, you take your ligand, you add it in a variety of different concentrations in these microcapillary tubes, and every time you have a ligand receptor interaction, it changes the environment of the fluorophore, causing a spectral shift. Long story short, you can get binding kinetics. You can get an idea of KDs, or on-off kinetics, and then from there extrapolate what your KD is. So both through SPR and MST, we showed that these self-assembling peptides has significantly better dose-dependent binding to VEGF receptor 1 and VEGF receptor 2 compared to QK, which is a mimic of VEGF, vascular growth factor. Some of the work that we published now several years ago demonstrates, or at least illustrates very clearly, the degree of angiogenesis that we get within these scaffolds. So in the blue box is the self-assembling domain, right, which forms a fiber. In the red box is the angiogenic mimic QK that I was telling you guys about. And as I mentioned before, that's present on every single peptide strand. So there's very high epitope density, very high signal density within that fiber. Now these fibers entangle to form a hydrogel that you can make fluorescent, you can track, what have you. But when we inject these in vivo, I'm just gonna try to draw here. Yes. When we inject these in vivo, this entire region here, right, down to the bottom right, this entire region here was acellular hydrogel, was just a gel. And within 7 days, we noticed rapid infiltration of blood vessels. All these red circles here are alpha smooth muscle actin, alpha SMA lined blood vessels that are 15 to 50 microns in diameter. So these are microvessels, arterioles, venules, small vessels that are mature blood vessels. And we can very easily see them burrowing or growing into these hydrogels, as you can see in these transverse and oblique sections of blood vessels. What's also interesting is that we have very little fibrous encapsulation. I'm messing out this slide. But there's very little fibrous encapsulation within of the hydrogels, and these hydrogels degrade within a 2 to 3 week period. Okay, great. All right. So having had those initial successes, having shown the safety and preliminary angiogenic efficacy of these hydrogels, at least subcutaneously, we then explored a variety of different applications for these materials. One of the first applications, maybe not chronologically, but one of the applications that we looked at was diabetic and infected wound healing. So one of the startups that I created, now defunct and shut down because not every startup works. I can tell you many lessons. One of the startups that we had, we got SBIR funding from the NIH to look at diabetic wound healing, where we created wounds in STZ-induced diabetic rats, filled them with our angiogenic hydrogel, and showed that these wounds heal relatively quickly within 28 days or so. One of the challenges in the wound healing field, be it diabetic or infected, well, be it diabetic or others, or burn wounds, is that it's a highly saturated field. So having something that works just as well or slightly better than a competitor is not always the best solution. And sometimes marketing or other aspects are important as well. In addition to that, these peptides, as I was telling Ali earlier, these peptides are classified as drugs because they bind a specific receptor. They bind VEGF-R2. On the flip side, if we can make a version of this or evaluate a version of this without that VEGF binding domain, they would be classified as medical devices based on our interactions and discussions with FDA. Anyway, so we've done some work with diabetic wound healing. We've expanded this work right now, in fact, to look at infected wound healing, not just single microbe, but polymicrobe wound healing, because apparently one microbe is not enough to evaluate. We're also very interested in looking at, we've also been very interested in looking at how these peptide hydrogels can improve outcomes for traumatic brain injury. So we did a fluid percussion injury on the cranium itself. We injected hydrogel and we showed that we could preserve neurons and this is the section of the brain, we could preserve neurons and decrease neuronal death at the very least in these SAP-treated rats. I got to keep my species straight. Anyway, one of the other things we've been very interested in is synergizing existing porous biomaterials. So we've worked with a number of different groups, notably Jan Yang's group, formerly at Penn State, now at Wesley University in China, where they work on polyoctane diol citrate materials. These are polyester elastomeric sponges, kind of like PLA, kind of like PLGA, those kind of materials, right? Polyester sponges, which have micron-sized pores, hundreds of micron-sized pores. When we implanted these porous sponges soaked with PBS, we don't see very good tissue infiltration or integration. When we load in our angiogenic hydrogels just by centrifuging gel into a porous sponge, we see very good infiltration of tissue deep into the pores. And if I did a better job, I'd show you a zoomed in picture, but you'd have to take my word for it. These are about 2 to 500 micron thick implants. And all the pores within these implants, hundreds of micron sized pores, are chock full of tissue. In fact, they have relatively large blood vessels. And I'll show you some of that data, at least for the dental pulp regeneration work on the next slide. We've done quite a bit of work in cardiovascular and peripheral vascular disease as well, where we've delivered these hydrogel scaffolds plus minus cells into the heart and into the limbs to recover from ischemia or low blood flow in the tissue. And some of the work that funds the lab, which I love to talk about, is our work in dental pulp regeneration. So right now, if you're a kid and if you need a root canal because your tooth got injured or you've got an infection, especially your permanent teeth, right now they put in rubber rods in a typical root canal that's called gutta percha, but your root, the part that goes into your gum line, is not fully stabilized. So the tooth can chip, crack, and eventually might get lost relatively easily, relatively quickly. What we propose is an angiogenic hydrogel that can help regenerate the dental pulp so that you have living tissue within the tooth that can further support root growth, lengthening, thickening, and apex closure. Long story short, we want to mature the teeth because otherwise these kids' teeth will fall out. So to that end, we did some work in canines and dogs and beagles a couple of years ago that we published in Acta Biomaterials in 2021, where we did root canals in canines. We injected the hydrogel into their teeth, and we saw beautiful dental pulp regeneration all the way from the apex, which is the bottom that goes into the gum line, all the way to the crown, the top of the tooth. And specifically, we saw an H&E, hematoxin and eosin staining, very nice and aligned tissue growth, as well as cellular infiltration into the regenerated tissue. If you follow this part here, which is in apposition to the dentin is the hard part of the tooth below the gums, enamel above the gums, right? And enamel surrounds the dentin. Anyway, if you follow this dense tissue border here, or regenerated tissue border here, we see that we have differentiation of the infiltrating cells that stain positive for dental sialoprotein, which is a marker for dental pulp-like stem cells and cells that can support root formation. So we also see that these cells start to have these finger-like protrusions that go into the dentinal canals, anchoring the cells and supporting maturation of the dentinal walls. We did Mason's trichrome staining and a bunch of other staining as well, but in Mason's trichrome staining, we see these nerve bundles that start to form. That's a 100 micron scale bar, so you can imagine that this is a relatively large canal, a couple of 100 microns wide, and this is an oblique section of a blood vessel. I love this drawing tool. I should do every presentation like this. We have these oblique sections of blood vessels which have regenerated, which are growing into the tooth, obviously carrying several blood vessels, carrying several blood cells. A lot of technologies, a lot of techniques out there that try to target therapeutic angiogenesis form capillary-like blood vessels that very quickly resorb within about a month. We're very excited because we form relatively large blood vessels that stay persistent in the tissue for several months after the hydrogel degrades. Some of the equally exciting, all students are doing great work. Some of the equally exciting work that we're doing is in hind limb ischemia or peripheral artery disease. So this is a disease that affects 1 in 5 adults above the age of 55 and probably younger too who don't eat well. This is caused by poor circulation in the legs, atherosclerosis in the legs, and ultimately this leads to severe muscle pain. Now stents, as I mentioned earlier, stents, grafts, endarterectomy, what have you, for larger vessels works great at helping reperfuse the tissue, the distal tissue. However, for microvasculature, so for small blood vessels on the capillary scale, arterial scale, venule scale, there is no FDA-approved clinical therapy to regenerate those vessels. There have been a number of attempts using stem cells, but engraftment is an issue. Survival is an issue. Using growth factors, either bolus growth factor therapy, gene therapy, mRNA nowadays as well, but nothing has made it past phase 2 or some phase 3 clinical trials. There's no product on the market. So we asked the question, can we develop a off-the-shelf scaffold? Can we develop a hydrogel that intrinsically stays localized in the muscle tissue in the calf that we can inject into the calf in multiple spots, like that first cartoon, that promotes blood vessel regeneration, especially microvascular regeneration? I'm going to walk you through this very quickly, but these are images of mice legs where you can, and this is laser Doppler perfusion imaging, where it's kind of like a heat map. More blood flow is red, less blood flow is blue. We tied off the ischemic limb, and we didn't tie off the non-ischemic limb. And what we see when we inject hydrogels into the ischemic limb after tying them off, or I guess the hydrogels up here, when we inject whole growth factor, we don't really see good restoration of perfusion. We don't see that much red going back to especially to the foot pad. If you look closely, there's bone exposed here, right? So this is a critical limb ischemia model. So it's not a gentle model. We tie off the femoral artery and the femoral vein. It causes severe ischemia, causing the limb to retract, the toes to amputate, and even oftentimes the whole limbs to fall off, right? So yeah, this was a lot of fun getting through the IACUC. But at any rate, so we injected the mimic alone. I told you about a couple of different mimics that we worked with, such as QK. We injected the mimic alone, and that too doesn't have good persistence, right? These growth factors, these mimics, these small molecules, they diffuse away very quickly, and they don't stay localized to the tissue site. And obviously, not obviously, but we injected saline as well, and that too caused severe limb retraction. And clearly you can see here the lack of digits and the lack of foot pad. What's interesting is that when we injected our self-assembling peptides, we've done a couple of different versions. I'm being very specific not to tell you guys, or not to let anybody know exactly which sequence, because we're filing IP on some of this stuff as well as other details. But some of the self-assembling peptides that we've injected into the limb have shown rapid restoration of perfusion, right? So this is before surgery in this picture here. This is right after surgery, 1 day after when we inject the peptides. And over time, we see very good restoration of perfusion all the way down to the foot pad, right? So you can imagine people who have peripheral artery disease, oftentimes they have diabetic ulcers that poorly heal. They have a lot of complications in their feet. Their digits, their toes oftentimes have wounds that don't heal well and require amputation, become necrotic and require amputation. So we're very excited that not only do we see good restoration of perfusion past the ligatures that we put in, the femoral artery and vein, but also down to the foot pad. What's also interesting is that we put them on a treadmill, and they ran at 28 days, a month later, they ran just as far as they did before surgery compared to any of the other groups that ran about 25% as much. And to be fair, they were running on 3 legs, which is still not bad. Not funny. All right, so that's the story on therapeutic angiogenesis. There's more work that we hope to do on that. And obviously, just because it works in mice, like a lot of other things, doesn't mean it's going to work in larger animal models in humans. One of the challenges with peripheral artery disease is that there is no good large animal model. The mouse model is the conventional model for demonstrating efficacy. However, a lot of studies that work in mice, as I mentioned, don't always work in humans, and that's been one of the challenges in that specific field. We are interested in pursuing, which is in part why we're very interested in pursuing some of the dental pulp regeneration studies, where microvasculature is also important, but the tissue volume and space is a lot smaller, and we can do large animal models like in pigs, which we're doing right now, to show efficacy in a large animal model before translating that to humans. And also, the clinical endpoints for a dental pulp clinical trial, the worst thing that can happen is you lose a tooth. The worst thing that can happen in a PAD, peripheral artery disease trial, is you lose a leg. So cost-wise, endpoints-wise, one's slightly easier than the other. Okay, so the next thing I want to talk about, I'm going to provide you a couple more vignettes of the work that we do in how we use self-assembly to address different clinical problems. I told you about how we designed a peptide that binds spike, right? So if you can imagine these red knobs are spike, we designed a peptide that binds spike. We call it spike binding protein, SBP, right? Yeah, we're creative. And then we attach a self-assembling peptide. And we showed through a variety of different studies that we can bind spike in a dose-dependent fashion with SBP3, the third version that we made. And when we add a self-assembling peptide domain, we see that it binds and self-assembles, like I was saying before, creating this fur ball on top of the viral particle, preventing it from interacting with host ACE2. One of the things that I want to show with this platform, if we ever get funded for it, one of the other things I want to show with this platform is that these peptide nanofibers we've shown get phagocytosed into both the cytoplasmic space as well as into vacuoles. So can we use this as a platform to essentially as an adjuvant of sorts, a platform to coat viral particles, get them to get phagocytosed and potentially present on MHC in differential manners. So one of the other things that I've been very fortunate to have are excellent collaborators. So we've worked very closely with Peter Krall over at University of Illinois, Chicago, where he does, where together we do molecular dynamic simulations where we take these spike binding proteins and put them on our self-assembling peptides, which form these beta sheets, right? So, and then we put them in proximity to spike or a variety of other domains, be it bacterial membranes or angiogenic receptor, VEGF-R2 or what have you. And we show that it binds to these different receptors and has this super stoichiometric or multivalent binding capacity. In addition to that, it makes very nice cartoons that look like this, which I don't know, I really like. Right. Anyway, so one of the last thing I'll mention about this paper that we published earlier this year is one of the interesting facets where even if we don't add a spike binding protein, that SBP thing, if we add just the self-assembling peptide scaffold, we still see that it inhibits viral infection of mammalian cells. And in part, that's because, or at least we think that's because there is a high density of electrostatic interactions between spike, which is highly positively charged, and this particular peptide, which is highly negatively charged. But that's an interesting phenomenon that we're exploring some more. All right, so the last thing I'll tell you about, one of the last things I'll tell you about is our work with not antiviral peptides, but antimicrobial peptides. We published a paper a couple of years ago where we saw that, unsurprisingly, that if you add lysines, if you have polylysine, it's antimicrobial because polylysines have a lot of positive charge and they interact with bacterial and fungal membranes, destabilizing them. And what's really interesting is that we've expanded this platform and seen that we can attack a variety, that we have efficacy against a variety of different bacteria, more than just some of the initial bacteria that we tried in that paper. And through a variety of different molecular dynamic simulations, we see that not only do we cause buckling of the membrane, but we also cause pore formation within the membrane, which we're trying to substantiate with TEM imaging. And then, of course, you got to take some nice SEM pictures, false color them because it looks nice. We're effective against a variety of different planktonic, so floating around bacteria, as well as biofilms. And that's been a big challenge with a variety of different antibiotics and even cationic antimicrobial peptides. One of the challenges is that even if you can kill a bug that's floating around, when that bug has secreted a calyx that's protecting it from the environment, it becomes very challenging for conventional materials or conventional drugs to inhibit their growth. Now, one thing that I like telling people about this is that we think that the mechanism of action here is a mechanical mode of action. It's kind of like a long, positively charged spear piercing through the negatively charged bacteria. So we think that because it's a mechanical mode of action, it's much harder to evolve resistance mechanisms, which is why we see, or at least which is why I think we see efficacy against multiple strains of a variety of different bacteria and fungi, as well as efficacy against biofilms. So this is actually a technology that we're very interested in moving forward towards the clinic. One of the things that I've learned in many startup failures is trying to translate a drug has many challenges. One of which being the very high cost of drug translation, as well as the high risk / high reward of translating a drug. And oftentimes, at least in my opinion, you need pharma to have a problem in order to make sure that they partner with you from an early stage to do those phase 2, phase 3 clinical trials. Otherwise, they are typically prohibitively expensive for regular old me to do a $100 million phase 3 clinical trial to take a product to market, take a drug to market. So I took a step back, and we asked the question: are there any medical devices that we can translate, preferably one with a 510(k) approval pathway? And one of the benefits of self-assembling peptides, at least beta sheet self-assembling peptides, is that there are predicates on the market. There's a company called 3D Matrix that makes a product called Puramatrix. I don't know if you guys have heard of that. It's out of a lab at MIT, Shugang Zhang's lab. But basically, it's like a 3D hydrogel to grow cells in. What we've seen is that our antimicrobial hydrogels and our hydrogels in general can help wound healing relatively efficiently. And we're interested in using this hydrogel platform for a variety of different indications. So while we're initially focusing on dermal wound healing, and we've got some large animal pig studies that we're doing. Not our pigs. My student is still working on the iACOOK. It's been like 2 months. I'm very proud of him, but it'd be nice to start these studies from the internet. Yeah, while we're focused on dermal wound healing, we're also interested in expanding this platform for a variety of other indications, especially through collaborations, both in the dental space, surgical space, and for bacterial and lung infections in the lung and brain. Bacterial and fungal infections in the lung and brain. So we've made a little bit of progress on this. As I was telling Ali earlier, I think the next company I make, I'm going to try to have all my ducks in a row, make sure we have some funding lined up, make sure we have our interactions with the FDA done, have all our ducks in a row before we start, and then immediately hit some milestones. We've done a Q-sub with FDA. We spoke to them a couple of days ago. Yeah, go for it. I'll stop.
I just wanted to ask the difference between the 3D hydrogel and regular hydrogel.
So 3D Matrix is a company that makes hydrogels, and it's like 99% water, 1% something else. And you can make these hydrogels any dimension, any shapes you want. Typically, they're 3-dimensional gels. Yeah. So just to finish up the slide, we've done a Q-sub. We are currently in the process of doing 510(k) enabling studies, which require money and GLP facilities. So we're trying to raise money or write a SBIR for that. We've identified a predicate. We have an idea of the studies that we need to do to get on the market just as quickly. It's called Puriderm Gel. And one of the things that I don't think about as a professor, one of the things that I don't think about at all really is how are you going to deliver this, right? Are you going to put it in a syringe? Are you going to put it into a spray? What size spray? What bottle spray? What's the bottle made of? Because all of these are important considerations that the FDA really, really cares about when you market a product, right? Like what's the format it's going to come in? Is it going to be in a needle? What size needle? What kind of syringe are you going to use? What kind of stability are you going to do on the final product, and things like that. So those are interesting aspects of medical device development that I too am learning now. And like what Allergan has done with Botox, where they have made a platform out of a drug, we're interested in seeing whether we can make a platform out of these self-assembling peptide medical devices for other indications as well. So very quickly, I'm going to summarize our approach in our lab at least. We take a look. So we started to do a lot of in silico design, where we take a look at known interactions of either through cryo-EM, X-ray crystallography, or solid-state NMR, solution-state NMR, interactions of different receptors and ligands. We then isolate specific regions that do the trick, that mimic the entire growth factor or ligand. We then, because we do self-assembly, we then attach a self-assembling tail. We make self-assembled peptides. We do bioanalytical studies to study structure, function relationships. We then do in vitro testing to demonstrate efficacy. We do some in vivo testing. This specifically shows a golden Syrian hamster model that's used for SARS-CoV-2. We don't have BSL-3 at NJIT, so we collaborate specifically for that. But we do run a number of other animal models, and the hope one day, get this in the clinic. We do a little bit of work. So since we started doing work in computational peptide design, we've also started to do a little bit of work in computational drug design. The sigma-2 receptor, TMEM97, I believe it's what it's called. The full structure of that was recently elucidated and published in Nature, I think in 21, 22. And since then, we've been a part of a consortium to discover novel ligands that can bind this sigma-2 receptor, which might be partial agonist or antagonist, different story, which may be beneficial in analgesia as well as treating the opioid crisis in some part. We've been interested in multiple sclerosis as well as a number of other diseases in not just targeting enzymes or receptors, but also potentially binding antibodies, which we think is a novel target to target at least. A lot of people are working on GLP-1 agonists, and we're interested in that as well. But instead of using linear peptides with alkyl chains to stabilize them on plasma membranes, we're interested in potentially using cyclic peptides, where the benefit is that cyclic peptides have more rigidity and conformational stability in binding targets. Building from that, we're working on cyclic peptides to bind complement 5A receptor. We do a little bit of work in drug delivery, so using adding a variety of different small molecules into these hydrogels that are aqueous-based, delivering them into the tissue for localized drug delivery. And we do some collaborative work on cartilage regeneration. All right, almost done. So I'm an engineer. I care about making stuff. So one of the things that we did was to make a toothbrush with vacuum on the back of it. So this is especially important for patients who have aspiration pneumonia, who have trouble swallowing, especially geriatric patients, some children, where we have a toothbrush head with holes laser drilled into them, and it sucks into a cavity at the bottom into a reservoir, basically a toothbrush with suction on it. Apparently it doesn't exist, and apparently it's a big enough problem that, you know, we got some money to do this. But long story short, I don't know if the best, I don't know translationally how much traction we've gotten. I do know, and we haven't gotten that much traction on marketing this product, but I do know that small aspirates or droplets of water are a big problem in space. Maybe we can get some astronauts to use this, you know, they get water droplets everywhere. We have a microwave peptide synthesizer in the lab, which is a huge boon for us. We make, you know, hundreds of milligrams of peptide in relatively short order. It takes about a day to make a 100 milligrams of a 20 mer peptide, and another week to purify it. But, you know, at least we get the synthesis done. But one of the issues that with the peptide synthesizer like that is that you have ongoing running costs as well as a fixed cost of about $70,000-$80,000 for that guy. That was my major equipment purchase when I started 8 years ago. So one of the things I asked my students to do was to miniaturize this so that, you know, those labs just starting out can make you know, 10 milligram scale of their peptides using a miniaturized peptide reactor. So that's another little project that we have going on. So very quickly, I'd like to think we're like a mini pharma company in a lab. Maybe not that much, but we do have a couple of supercomputers doing a lot of our computational work. We use NSF Exceed as well as the NJIT cluster called Wolver to run a lot of our different simulations. We do synthesis as well as in vitro characterization, a little bit of 3D printing, material characterization, the SEM, TM, and AFM in our core facilities, but otherwise we process samples in the lab for those at least. We do stability, cell culture testing, both microbial, bacterial, fungal, as well as mammalian cell cultures. We do a variety of different binding assay characterizations using SPR, Western, microskeletal morphophoresis, MST, synthesis and formulation of materials and peptides, and then of course a whole host of different animal models, in a vivarium. The last thing I'll mention is that science can't be done in silos. I don't think any of the successes I have had to date or any of the papers I published to date were just me. I know that. I think collaborative research is extremely important. And as I mentioned to a number of different extremely talented and exciting faculty that I've met here today, I'd love to carry on this tradition. I'd love to build collaborations and both submit grants and publish some really cool papers on how we can integrate some of these self-assembling peptides, some of these materials and technologies into your projects as well as what we do. Specifically, you know, we're interested. I told you about how when we self-assemble, all those mimics are right next to each other, so it kind of messes up the self-assembly. We don't get very good gels. So we've come up with a new peptide system where we can attach the mimic domains orthogonally right, perpendicular to the direction of self-assembly. So you got this brickwork, and then you got the mimics coming off perpendicular to it. Sequence is not there because we're trying to file IP on it and other details that I'm not going to go into. We're very interested in expanding our work in periodontal, endodontic models. So if you guys have materials technologies that you're interested in working with us, you know, we have those models up and running in rats and pigs. But rats is easier, pigs more complicated. We're also interested in both prophylactic and therapeutic utility of antiviral peptides, but more specific to what we're doing today is translation of these antimicrobial, antifungal, antibacterial peptides, which are efficacious against biofilms as well as planktonic species. So here are a list of collaborators. I'm sure I'm missing a decent number of them, but we're very excited about the collaborative work that we do. We've been very fortunate to get a decent amount of money. You know, a lot of this is collaborative funding. We've been very fortunate to get a decent amount of money, especially from the NIDCR, as well as a number of other NIH institutes. And yeah, so right now we've got me, we've got Corey, who's a research assistant professor in the group, Laura, who is a postdoc. She recently had a baby. And we've got 5 graduate students, Abhishek, that does a lot of our animal work, Bobak, who does a lot of our in vitro work, Gelavis, who's a trained veterinarian, so she does all of our animal work along with Abhishek, but she's like the surgeon doing it. Alexandra Griffiths, she does a lot of our dental work, and Joe does a lot of our computational work. Here are 4 of the undergrads who weren't running around doing other stuff. Here are 4 of our undergrads, and the lab, we have a variety of other undergrads as well. So yeah, thank you very much.
Thank you, Dr. Kumar, for the amazing talks. Comments? Yeah.
I was just wondering, for the self-assembling peptides, if you were to use them in other things, like other than for other things other than angiogenesis, would they have like a different domain for that specific thing that you're trying to?
Yeah.
Each time you create the hydrogel with the self-assembling peptides, like depending on what it's for, they would have
a slightly different sequence. Yeah. So that's a good question, and that's been a challenge because we've published on a variety of different applications with different mimics that we've attached to the self-assembling platform, and we've learned a lot of design rules along the way through making many mistakes. And in part, it's because sometimes they don't solubilize, sometimes they don't self-assemble, sometimes the mimic is too large. We need to tweak the self-assembling sequence one way or the other. So yes, the easiest example is we typically put lysine residues on the termini of these peptides. However, depending on net charge, we might switch them out to glutamic acid or other charged amino acids. Off late, we've changed even the self-assembling domains to incorporate phenylalanine, for example, to enhance pi-pi stacking or other kinds of interactions for self-assembly. But yeah, it's a good question. Every peptide is slightly different.
Sorry, I had one more, just to clarify, the way that it worked with the COVID was that the self-assembling peptides like wrapped around the virus and blocked the spike protein from binding to human cells?
Binding ACE2, yeah, binding human cells.
Okay.
Yeah, exactly.
Thank you.
Epiviral self-assembly.
So I have a question regarding this actually. So we're talking about like that gel is being an adjuvant, and you're thinking that it can like the spike protein or peptide derived from the spike protein could be loaded on MHC2. But my question here is like, if it's binding super strongly to the spike protein, how is it going to be actually degraded inside of there and actually be released to be presented on class 2?
Right. So let me put it this way: these self-assembling peptides have affinity to each other because of the hydrophobia interactions, van der Waals interactions, supramolecular interactions. They also have affinity towards their targeting domain because of complementary interactions, right? Now, the degree to which one is greater than the other, we don't quite know. But the hypothesis, and at least what we've seen through some imaging, is that these peptide nanofibers these peptides form fibers that get phagocytosed, and what we see in the vacuoles are short fibers, right? So there is an on and off kinetics that's always happening. So that's one part. My hope, now that we've shown this, my hope is that we actually see, oh God, where was it? Okay, hold on. Yes. My hope is that we can actually one day prove this, right? That we can show that the peptides code SARS-CoV-2, and then that whole construct gets phagocytosed and then sliced and diced and then presented at AMHC. However, every grant that I've submitted to NIH for SARS-CoV-2, they've told me show efficacy for MERS, SARS, and SARS-CoV-2, and show animal efficacy before you apply. I'm like, I don't even know anyone who works on MERS, let alone SARS-1, right? Or SARS-OG.
So this also comes back to my second question is, you're saying that like when it phagocytoses, it's in shorter peptides. So are those peptides immunogenic? Because also you're talking about, maybe blocking antibodies. And so again, if an antibody can recognize some of those, did you test any immunogenicity?
Right. So I do not have that data here. Okay. So we've done, sorry, I don't mean to scroll through all of this. So doing immunogenicity testing is expensive. So for our medical device that we plan on trying to translate, we've taken these gels, we've injected them every 3 days for 30 days subcutaneously in mice, and we've seen no increase in IgG, IgA, IgM, IgB, any of the Igs, right? So we've shown that these peptides are short enough that they don't typically, like most other short peptides, don't elicit adaptive immune responses. In subcutaneous boluses, right, when we inject boluses of these hydrogels into the tissue, I'll just show you this. Within 7 days, we see a lot of cells and tissue, different types of tissue that infiltrate, but we also see a lot of cells that infiltrate. And the DAPI staining in blue here, if you were to co-stain that with a lot of those would co-stain for CD68 or neutrophil markers as well. So one of the things we're trying to figure out now, which suggests there's a strong innate immune response. However, we don't see fibrous encapsulation either. So it's a highly localized response only to the bolus. And when we take this, embed in EPINATE section for TEM, we can see that those cells that are infiltrating are phagocytosing the peptide hydrogels, degrading it, right? So ultimately, I guess the question is, or rather what Bobak, one of my students, is doing right now is he's injecting these hydrogel boluses, taking them out at different time points and running FACS to understand what are the different types of cells temporally that go in and whether we can tune it based on the mimic that we add. Thank you. That's the most questions I've gotten.
For your COVID-19 project, did you compare your peptides to using ACE2 decoys that can prevent the binding onto spike protein?
Yeah, so, I mean, this is kind of like an ACE2 decoy, right? Did we use, I don't think we did. I don't think we used ACE2 decoys. I don't remember, like, okay. So one of the, I think it was Johnson or one of the faculty I was speaking to earlier, research is not always done linearly, right? You make something, you're like, oh, this is really exciting, this is really interesting. Let's try it in an animal. Let's try it in the money experiment, right? And you show that it works, and then you work backwards. So I don't remember exactly what control we used. I do know at some point we used convalescent plasma as well as one drug. I don't remember which drug, so I apologize. But it's in the paper, which I could look up, but I don't remember. But I don't think we used specifically ACE2 decoys as a control. Because that was done in 2020 when we came up with 2021, when we came up with the idea, applied for funding and got it, and started doing the work. And as you can see, this was published like in April of this year. Because that's how long it takes. Long it takes.
More questions?
So first off, quick clarification. For your hydrogel that is angiogenic, or that you demonstrated angiogenesis with, Did you say that that is it has shear thinning properties?
Yeah, yeah,
And I imagine you've tested the injectability of it, right?
yes.
Have you tested anything to do with 3D printing or 3D printing this gel?
Okay, so these are shear thinning and shear recovering hydrogels. When you make them at high enough concentrations, they stand when you squirt it out through a syringe. We specifically haven't 3D printed them because, you know, two hands with many hands in the lab. But others have, and it 3D prints well.
Cool. As in, like, it's freestanding, or
Yeah,
you like it?
yeah. In fact, there's an advanced materials paper by Jeff Hartgring that was published earlier this year, I think. I'm pretty sure earlier this year, maybe last year. It's in advanced materials. It's 3D printing of self-assembling peptide hydrogels.
Cool.
And they show not this sequence, but a similar sequence, how if you have it at high enough concentrations, you can 3D print it. I would be very interested to see if these specifically 3D print, but I think that's a question of optimizing concentration, formulation, buffer, what have you. But they form gels, so I mean, I don't see why you can't shear thin and print these gels as well.
Out of curiosity, have you seen whether anybody's tested 3D printing of similar gels into some other medium, for example, rather than having it freestanding on its own, but be supported by something else?
I refer you to the Jeffrey Hartgring paper published in Advanced Materials, sir, because I don't remember all the details of that.
Sorry, just why you mentioned that for the peripheral neuropathy, you mentioned that you only did the treatment for mice, and you don't know if that would work in humans. What was the reason, like, you can't do it on pigs?
Okay, so if you tie off the femoral artery and femoral vein in a human, that leg's going to fall off, or anything larger than a mouse. So a mouse's femoral artery and femoral vein are on the order of about 100, 200 microns, right? But in a rat or in a mini pig or a pig, it's on the order of millimeters. I don't know of a technique that regenerates millimeter-sized blood vessels, right? So we're regenerating blood vessels on the tens of microns, maybe 100 micron diameter size, right? So in terms of vessels that are being regenerated that are comparable in size to a mouse, that are comparable in size to what's required for treatment, a mouse is about the same size, right? In a human, I wouldn't inject these in the femoral artery. I would use a vascular graft or a stent or an established technique that vascular surgeons use. I would inject this into the muscle where I have a lot of muscle pain, right? Like these people are just sitting down and like they walk like 20 feet and their muscles hurt like crazy, right? It's called intermittent claudication. So for them, they have these small, their vessels, their microvessels are stenosed, are hardened, have plaque in them. And those vessels, again, are around these 200, 500 microns. So the model to show that it kind of works there would be the high limb ischemia model in a mouse. Yeah.
Couple of other questions in online. The first question is, do you think the computational result align with the experimental findings? If so, in what aspects?
So this goes back to your question, which is sometimes you make something, you do the experiments and you see that it works, and you come back and you do the computational work to help explain why it works, right? To provide some buttressing or confidence for that. I would like to think that we're at I definitely think that we're in the direction, by we, I mean the field community is in the direction of rationally designing materials and peptides, at least peptides and proteins, in a computer that have a pretty decent chance of working once you synthesize them. Now, we take the extra step of adding a self-assembling domain, and that adds a lot of complexity and is really throwing a wrench into that whole design paradigm. However, I would like to think that we're getting closer to refining our techniques to come up with better guesses. End of the day, a computer is the computer is just guessing what we think is going to work, and we would be very happy if it made those guesses well. When we do these, we come up with hundreds, if not thousands, or more sequences that we then use different criteria to limit down to like 5 or 10 that we actually synthesize and test.
Okay, thank you. Thank you, everyone. And thank you,
Thank
Vivek.
you.
Give a big
Happy to be a resource. Bye. Awesome. Thank you very much for your talk. Thank you.